My dear students, today I am continuing with the second video class in the chapter Chromosomes, and in previous chromosome class we have seen the number of chromosomes, the structure of chromosomes, classification of chromosomes and fine structure of chromosomes. And in the fine structure of chromosomes we have seen three models: folded fiber model and multiple fiber model and nucleosome model. And nucleosome model is accepted and it is true till today. Up to this we have studied, and apart from this we are observing one more important aspect here, that is, in some of the organisms different types of chromosomes are existing. Very big sized chromosomes are existing, and such chromosomes are called as giant chromosomes. Giant chromosomes. So these are called as giant. The word giant stands for big. Giant chromosomes are big sized chromosomes are present, and such chromosomes are called as the giant chromosomes. And the giant chromosomes are of two types. For the study, we are classifying them into two types. One is called as polytene chromosome. First one is called as polytene chromosome, and the second one is called as lamp brush chromosome. Two types of these two are giant chromosomes only. What do you mean by giant? Giant chromosomes means they are bigger than the normal sized chromosome. What you are observing in the normal chromosomes, so they are bigger than that. Sometimes you can observe them with your naked eye. Without the help of microscope, you can observe them. They are such a big sized chromosomes, and such chromosomes are called as giant chromosomes or the abnormal chromosomes. And these giant chromosomes are of two types. One is called as the polytene chromosomes, and second one is called as lamp brush chromosome. Let us see one by one. What do you mean by polytene chromosome? First we shall study polytene chromosomes. So polytene chromosome means what? The name itself indicates the meaning of the polytene chromosome. Poly stands for many. Teen stands for threads or DNA fibers. Usually, yesterday we have seen the structure of chromosome. In the interphase, the chromosomes will not be found in the form of rod-like structures, but they appear in the form of network thread-like structures, and that is called as chromonema. But each chromosome is made up of only one chromonema. But here, in this giant chromosome, so each chromosome is made up of many chromatin fibers, chromonemata. So because of that, it is called as polytene chromosome. In other words, the normal chromosome is unitene, but it is polytene chromosome, and these are found in the salivary gland cells of Drosophila, melanogaster, and some of the lepidopteran insects we are observing, and they are found chiefly in the salivary gland cells. Salivary gland cells, they contain the nuclei, and those nuclei are containing the polytene chromosomes, and chiefly they are found in the larval salivary gland chromosomes. So what is the shape of this? So the shape of the polytene chromosomes is very interesting. So in Drosophila, yesterday we have seen n equal to 4 and 2n equal to 8. 8 chromosomes are diploid set and 4 chromosomes are the haploid set. But in this case, you can observe all the chromosomes are the maternal and paternal, two sets are present in that, and all the chromosomes are joined end to end. From one end to the other end, they are united. And the chromosomes, they are 8 in number. The centromeres of all 8 chromosomes are united. They are fused to form a central mass, and that is called as the chromocenter. It is not called as centromere, it is chromocenter. Let us see now what is the structure. So this is called as the chromocenter, and we are observing the structure like this. So this is one arm. So this is one arm. This is one more arm. So, and you will find one more structure here. One more structure. And apart from this, you will find one chromosome here, and you will find one chromosome which is very long. So, here you can observe, in this one there are two chromosomes. So, you can find here this particular thing, and this you can call third chromosome, or you can call this is the first chromosome, and this is the second chromosome, this is third chromosome, and this is fourth chromosome. Okay? But in the beginning I have told you there are 8 chromosomes, but we are observing here only 4 chromosomes. So I will just draw the diagram here to clarify the doubts. So this is the chromosome, and this is called as chromocenter. And attached to this there will be a nucleolus. This is called as nucleolus. So here, third left arm, third right arm. This is fourth left arm, and this is fourth right arm. So you can see here, first chromosome, second chromosome, and you will find one more structure here. One more structure. So this is X chromosome, and this is Y chromosome, and this is X chromosome. So like this you are observing this one. But now you can ask me one question very simply, that sir, you are telling about 8 chromosomes in the diploid set, but you are observing only 4 chromosomes. This is one chromosome, this is another chromosome, this is one chromosome, this is X and Y chromosome. 1, 2, 3, 4. So four sets are there here, four chromosomes are there. But actually this particular thing is containing 2 chromosomes. So this is maternal set and this is paternal set. From this side to this side they are united. And that is called as the third chromosome. So if you stretch like this, it will become one chromosome, but which is containing the diploid set. So this is called as the maternal, and this is paternal. But here the fourth chromosome is also having the 2 chromosomes here. So for example, if I make this one shaded area, so this is one chromosome. Okay. So this is one chromosome, and unshaded area is one more chromosome, and this is called as the fourth set. And here you will find the shaded area, so that is one chromosome. So you can find here shaded area, and here also this is shaded area. So first pair, second pair is X and Y. So here 2 chromosomes, here 2 chromosomes 4. Here 2 chromosomes 6, here 2 chromosomes 8 chromosomes are there. So one speciality of this one is this contains many chromonimata. Chromonimata are many, and they from one end to the other end, the 2 chromosomes, bivalents what you can call, so they are fused, and the centromeres of all the chromosomes are united here. It is called as chromocenter. And if it is male Drosophila, you will find one big, large Y chromosome and a very small X chromosome. This is called as X chromosome. And if it is to be female, so Y chromosome will not be there, you will find 2 X chromosomes. Anyway, so for the reference I have taken the male Drosophila, so you will find the structure like this. I will repeat once again. In Drosophila melanogaster, the what you call the salivary gland chromosomes or polytene chromosomes are containing 8 chromosomes, and first pair, second pair, third pair and fourth pair. Four pairs are there. But in each pair, there are 2 types of chromosomes. One is maternal and paternal. Maternal and paternal in normal case, they are away from one another. Only during the meiosis, they come closer for synapsis. But in this case, salivary gland chromosomes, they are always united. They are always fused. And one thing, when the mitosis takes place, so there is no separation of the chromatin fiber, they remain inside and what is known as endomitosis, and because of that you will find the chromosomes are containing many threads, many chromonimata, and because of that they are called as polytene chromosomes. And because of many number of thread-like structures present here, the size becomes big, the chromosomes become big sized, and because of that they are called as the giant chromosomes, and because they contain many chromonimata, they are called as the polytene chromosomes. Now coming to this one, you will find many fibers like this, you will find like this. So if you observe under high power, you will find like this. But here you will find some thickened areas, and so somewhere you will find unthickened areas. They are called as the chromonimata. And chromonimata, see, you will find the thick area, and these are called as chromomeres. They are called as chromomeres. But if you observe carefully, here, you will find the structures, what is known as, say here, you can find here, I'll draw here only. So here you will find some balloon-like structures existing here. So you can find here balloon-like structures, and these are found in the interband area. So you will find the band-like structures here, bands. So for example, if you see the chromosome here, you will find the dark area like this, and this is called as band area. This is called as band area. The area present between 2 successive bands is called as interband area, and in the interband area you will find innumerable balloon-like structures. Innumerable balloon-like structures, and these balloon-like structures are called as Bulbiani rings. Bulbiani rings. So what do you mean by Bulbiani rings? Bulbiani rings means for the first time such type of rings are observed in the interband area. So this interband area shows innumerable balloon-like structures, and this was first time observed by a scientist called as Bulbiani in 1881. So he first for the first time observed them, and he discovered them in order to honor the scientist. So these balloon-like structures are named as Bulbiani rings. And what is the function of Bulbiani rings? Bulbiani rings are the sites of rich protein synthesis and rich RNA content. They are highly active sites. So such things are present in the polytene chromosomes. Okay. Now coming to another type of chromosomes. So that is called as what is known as lamp brush chromosomes. They are called as lamp brush chromosomes. These are found in the oocytes of the oysters and some of the reptiles and some of the amphibians you will find, and such amphibian oocytes and reptiles, they show this type of big sized chromosomes, and such chromosomes are called as lamp brush chromosomes. What do you mean by lamp brush chromosomes? So in the normal case, lamb breast chromosomes are having the structure of the brush which is used to which is used to clean the lamps, the glass covers of the lamps in the olden days. So they appear like this. Such structures are called as lamb breast chromosomes. They are very big sized. So such structures are present here. So these are found in the meiotic stages. Meiotic stages they are found. So meiotic stages they are found. They appear during the diplotein. You know, in meiosis, it has got a prophase 1, metaphase 1, anaphase 1, telophase 1. Prophase 1 is having the stage what is known as leptotein, zygotein, pachytein, diplotein, dikinesis. But these particular chromosomes are observed during the diplotein of the meiosis. So during this, you will find this is called as the main axis. Main axis. And these are called as puffs. Puffs means loops. They are called as the loops or puffs. You will find loops or puffs. And these loops, they are also called as loops. So innumerable loops are there, and such things are very big in size. Sometimes you can observe with the naked eye without the use of microscopes, and such big sized chromosomes are called as the lamb breast chromosomes. These are found in the oocytes of reptiles and amphibians. Not only that, they appear like this only during the diplotein of the meiosis 1, prophase 1 of meiosis 1. So if you observe this particular thing here, so this is the structure, you will find like this, and so this is called as the loop. One loop I have enlarged here, one loop, and this loop is a very active site, just like the Bulbiani rings in polyethylene chromosomes. These are the active sites of protein and RNA. Such loops are called as, so they contain the matrix and they are rich in protein plus RNA synthesis, and they are active sites. So here the RNA synthesis is more and also protein biosynthesis is more. Such loops help the organism for the synthesis of protein, and such particular things are found chiefly in the oocytes of the amphibians. Okay. So with this, only two types I have told you already. So these are called as lamb breast chromosomes and polyethylene chromosomes. And apart from this, I will just give an idea, not in detail. There are special types of chromosomes. Apart from normal and giant chromosomes, special types of chromosomes are there. The first one is called as supernumerary chromosomes. Supernumeraries. These are called as special types. Special types of chromosomes. They are called as supernumerary chromosomes. Okay. And supernumerary chromosomes means usually they are called as extra chromosomes. Sometimes the organism may contain in the normal set of chromosomes, they contain additional chromosomes. Such chromosomes are called as the supernumerary chromosomes. Sometimes they are called as B chromosomes. They are called as B chromosomes. So they are found in some plants like Tradescantia. And second one, L chromosomes. L chromosomes. What is the meaning of L? L means they are limited chromosomes. Limited chromosomes. These are large chromosomes and limited to certain individuals. They are not found in all organisms. They are particularly found in certain organisms. Especially in some insects, they are found. So because of their limited occurrence, so they are called as L chromosomes. And second one is M chromosomes. M chromosomes. M chromosomes. M chromosomes stands for minute chromosomes in the nucleus. Apart from the diploid sets of the chromosomes, sometimes we find the dot-like structures. They are not having the chromatid, centromere, secondary construction, primary construction. All these details are not there. They are in the minute nature. They are available without any morphology. And such chromosomes are called as M chromosomes. They are also found in certain insects, and they are not survive. They will not survive for longer time, and they become vanished. Okay, with this we have completed what is known as the special types of chromosomes, and next one is aneuploidy in humans. This is a very interesting topic. Aneuploidy in humans. What do you mean by aneuploidy? So here ploidy stands for basic set of chromosomes. For example, in humans, total 46 chromosomes are there. It is called as 2n. Okay, 2n is nothing but diploid. So this is having 2n. So then what do you mean by n? N means it is 23. It is called as n. So this n is nothing but the basic set of chromosomes available at any time in the organism, in human beings. Such particular number of chromosomes are called as ploidy, or basic set of chromosomes. So this is called as ploidy. In this ploidy has been classified into two types: euploidy, and another one is aneuploidy. Okay, aneuploidy. So what is the, of course there is a separate topic for this, that there I will discuss in detail, but now we shall understand what is aneuploidy and what is euploidy. Euploidy stands for, see here, n is there. If another n is added totally, for example 23 are there, n. If another n means all 23 chromosomes are added, so this will become 46. Okay, and if 23 plus 23 plus 23 are there, 3 ploidy are united. So this is called as 3n. This is called as 2n. Okay, so that is called as euploidy. Euploidy means addition of entire set of basic set of chromosome, or the ploidy. That is called as euploidy. So euploidy we are not interested right now. So aneuploidy means it is the addition of chromosomes, no doubt, but in this particular addition the entire set will not be added. Say for example 2n. For example 2n means 46 chromosomes are there. For this, one or two chromosomes are added or deleted, not the entire set. If another 23 set is added, that becomes 3n. That becomes ploidy, euploidy. But here the entire set will not be added, but only the part of the set is added or deleted. For example, if another chromosome is added here, what do you write now? 46 is nothing but 2n. So it is written as 2n plus 1. This is called as aneuploidy. For example, 46 chromosomes are there here, plus one chromosome is disappeared. In the basic set only one chromosome disappears. In that one what you write? 2n minus 1. Both are called as aneuploidy. If 23 are added totally, that is called as euploidy. Euploidy is not there here. We are studying aneuploidy in human beings. So what happens in human beings about aneuploidy? Let us discuss now. So first, aneuploidy stands for the addition or deletion of part of the set of the chromosome, or part of ploidy, that is called as aneuploidy. So let us start studying what is aneuploidy now. Aneuploidy in humans. Aneuploidy in humans. So in man, some of the serious things are happening due to the addition or deletion of one or two chromosomes. If one, see, normal chromosome number in human being is 46. If any person comes along with one chromosome extra, 47 chromosomes, so he will suffer from so many disorders. So now let us see one by one. So first thing what you are observing is Down's syndrome. Down's syndrome. Okay. In Down syndrome we write 2n plus 1. 2n plus 1. No, here 2n plus 1 is equal to 47 chromosomes are happening here. So the individual, the human being, which is containing one chromosome extra, not the entire set, one chromosome extra, 2n plus 1, that is called as Down syndrome. Why it is named so? This type of syndrome was first discovered by a scientist, John Down, in 1828. Because of that particular person, this disease is named after Down only. Down syndrome. So here, this particular person having this syndrome will have one autosome extra. Autosomal syndrome, we are calling this one as autosomal syndrome. 21st chromosome. So if you arrange the chromosomes into 23 category, 21st chromosome, each chromosome has its own paternal and maternal. But apart from that, there will be one more chromosome. So 21st chromosome has got in 3 sets. Usually all chromosomes are in 2. But here there are 3 chromosomes of 21 set. Because of that, the person is having 47 and it is denoted by 2n plus 1. Down syndrome. And the person having this Down syndrome is having so many disorders. That is, the person is very short and eyes are slanting and the neck is webbed and the face is very ugly and the voice is very crackle. So many things, and ultimately his IQ is very, very low and he is very dull and diseased. So all these so many disorders are there, and this particular syndrome person will not survive for many days. He will die. So this is called as Down syndrome, and it is in some other case it is also called as Mongoloid syndrome. It is also called as Mongolism. It is called as Mongolism. It is also sometimes it may be asked in the examination. Mongolism. Mongolism is nothing but Down syndrome only. Down syndrome, because this particular syndrome was first discovered by a scientist called as John Down in 1821. But this particular disease was first discovered in a person who is belonging to Mongolia, a country. So a nation. So where in Mongolia, so the person was belonging to Mongolia, and because of that this is also called as Mongolism. So 2 names of this for this particular syndrome. Down syndrome, because this was discovered by the scientist called as Down. It is called as Mongoloid syndrome because this particular syndrome was first noticed in a person who belongs to the country Mongolia. So this is called as Mongoloid syndrome. Okay. Then coming to second disease, first of all, what is a syndrome? We have to understand, it is not a disease actually. So it is a person containing group of diseases is called as syndrome. For example, AIDS we are calling acquired immunodeficiency disease. It is not disease, it is syndrome. So syndrome means a person who is having AIDS, he will have all the diseases. He is the person, a walking museum containing so many diseases. So since a group of diseases are present in that person, the person is called as a syndromic. Here also, so many others, disease symptoms are available. Because of that, it is called as Down syndrome. So another one is called as Klinefelter syndrome. Klinefelter's syndrome. This is also syndrome because, so Klinefelter syndrome, because This particular syndrome was first discovered by a scientist called as Klinefelter, Henry Klinefelter, and because of that only the disease was named after that particular person. Klinefelter for the first time discovered this disease. So what happens here? Here actually normal person is what? 44 autosomes are there in our body and XY are there in our body. This is a normal person. All of us are XY, male, and 44 plus XX is the normal female. It is normal case. But sometimes due to some abnormality, the person is coming or person will be having one chromosome extra. One chromosome extra. So we don't confuse with this one. So this one XXY. So totally what happens? 2n plus 1. So totally 47 chromosomes are there. Now you can ask me, sir, here also 47, here also 47, both are same. No. Here the allosome is extra, here autosome is extra. So that is the major difference here. So allosome is extra and the autosome is extra. But here what happens? If two X chromosomes are there, the fetus develops into a female body. If X and Y chromosomes are there in the fetus, it develops into a male body. But here X, X, Y are present. What happens? It is neither totally male nor totally female. So the body structure, what you are observing is, the body structure is female body, but the tendency is towards male. So you can find here the breast development, broad chest, and shrinkle wise just like females, and so many other characters are observed, and on the chest the hairs will not develop properly. So this is the female characters, but the person containing this particular syndrome tries to live like male only. So many things we are observing in our nature. So that is, they are actually females, but they try to behave like males. Such things are called as the Klinefelter syndrome because of the presence of one extra chromosome X, XX. So these two X always makes the body female. But when Y is present, Y makes the body to be male. So this is neither male is complete nor female is complete. It is in between the male and female. That particular thing is called as Klinefelter's syndrome. And the last one is the Turner's syndrome. Turner's syndrome. Okay, Turner's syndrome. This is also a syndrome which is happening due to the disorder in the allosomes. Usually, what is the normal male? I have told you already, 44 plus XX or 44 plus XY. Normal female, normal male. But actually, in the Turner syndrome, what happens is 44 autosomes are there. Here also 44 autosomes are there. There is no problem with autosomes, but the problem is with the allosomes only. And because of the allosomes, what happens is only one X chromosome is present, neither Y is present nor X is present. Try to understand this. And the total number of chromosomes is nothing but 45 chromosomes. One chromosome is totally missing. It is XO. O means 0. So only X chromosome are there. So this particular organism or the human being will not be female. But actually it looks like female, but internally the ovary will not be developed and organs will not be developed. But actually there is a body, female body, but which is totally sterile, imperfect female body is there. And this particular disease was named after the scientist who has discovered this particular syndrome, what is known as Turner syndrome. So Turner is the scientist who has discovered this particular disease. So with this we have completed the study of chromosomes. So there are 2 video classes depending upon the chromosomes. In this class I have discussed giant chromosomes, that is polytene chromosomes and Lamprey chromosomes. See the structure and observe the notes what I have given in my WhatsApp group and also in the Google Classroom, and also try to go through or hear the video class slowly, carefully, and in spite of that, if you get any difficulty, so don't hesitate to call me to the phone number what I have given in the previous classes. So contact me and try to study. So next coming days I will give some more video classes pertaining to other topics. So have a nice time.